This is the third lesson in the series about excretion, and it's the second lesson when we look specifically at how urine is formed, focusing on the functioning of the nephron. For today's lesson, please make sure that you have read your textbook pages 175 to 179, and by the end of today's lesson, you should be confident in completing the notes on page 26 to 31 of your booklet. These are the learning objectives that we have remaining, but for today's lesson, we are going to focus on the top one, which is the process of filtration, reabsorption and secretion within the nephron itself. So when we look at urine formation, it has three phases, as we discussed in the learning objective, and broadly they are these three things. So the first one is glomerular filtration, and this is where blood is filtered under high pressure at the glomerulus, that knot of blood capillaries we talked about in the previous lesson. So because of this filtration, liquid is filtered from the blood and it enters the nephron tubule. All of that stuff that is in the nephron tubule is indiscriminately filtered, so we need to retrieve the things that we want to stay in the blood. So the next process after the filtration in the glomerulus is selective reabsorption. It's selective because we are choosing what we want to go back into the blood and what we excrete from the body. And the third process and the final part of urine formation is tubular secretion. So things that are added to the nephron tubule. So secretion is when we want to remove it from the blood and add it into the nephron itself. So just to review what we learned in the last lesson, we have a cross-section of the kidney again here. The outer area, of course, is the renal cortex, and inside we have the renal medulla. Then the medulla is separated out into renal pyramids and these renal columns. Within the kidney itself, we have these tiny filtration units called nephrons, and this cartoon here is showing that cartoon image. Now, within the nephron itself, we have the glomerulus, that knot of blood capillaries, and that sits inside the glomerular capsule. The first twisting and turning structure is called the proximal convoluted tubule, which goes into the ascending and descending loop of Henle. On the other side of the loop of Henle, we have the distal convoluted tubule, which connects to the collecting duct. Now, this is just a basic cartoon image, and the one that you've drawn before in the previous lesson should help you to make notes for this lesson. Now, there's a couple of things we want to look at. Now, we're going to focus on that first stage, which is glomerular filtration. Remember that this is going to be our renal artery, or a branch of it, and a branch of that renal artery, which is the afferent arteriole, leads into the nephron. And we did note that the afferent arteriole is wider than the efferent arteriole. So the reason why that becomes important is all to do with pressure. The afferent arteriole branches off from the renal artery and it brings blood to the glomerulus at the nephron, and that blood is under relatively high pressure. Now, as we've just said, the diameter of the afferent arteriole is much wider, and as it goes into the glomerulus, it starts to turn into the efferent arteriole, which is much narrower. Now, because we have got this narrowing effect going on here, it creates a pressure difference. So if we think about what we know about the relationship between the diameter and the pressure, you know that if you are going to spray someone with a garden hose, for example, you will actually squeeze that tube, that garden tube, and what you're actually doing is you're making the diameter much smaller. And when you do that, the water rushes out at much higher pressure because a lot of water has got to squeeze through a small space. And that's what's happening here. you have got low blood which is under high pressure from an artery, and as it goes from high to low volume diameter, the pressure will increase. Now, because we're increasing the pressure in the glomerulus, the blood is under pressure filtration, so the smaller components of the blood are squeezed out and we end up with filtrate leaving the blood and entering the glomerulus. So now what we have is the filtrate, which is known as glomerular filtrate. So this is process number 1. Now when we look at this process of filtration again, in this diagram again, we've got the afferent and efferent arterioles, and what's actually happening is a filtration effect. In this cartoon picture, we've got a picture of a sieve just to illustrate that. So we know with sieves, large molecules won't be able to get through, but smaller ones absolutely can. So this liquid is no longer blood when it's in the nephron, it's filtrate. So what is filtered out of the blood? Things that you must remember and what you must know as examples. Small molecules such as water, glucose, amino acids, urea, toxins, salt, sodium and chloride ions. Now, when we look at this list, it is quite a long list and you'll be able to identify that there's some good things there and some not so good things. So we've got to be thinking about, well, why is all this happening? Well, the reason for that is because filtration happens indiscriminately. All it knows to do is to keep big things in the blood and filter out the small things. What is left inside the blood are the big things, which are plasma proteins, the red blood cells, and some of these things that don't make it during this turnaround. So now we're looking at a diagram of the start of the nephron. You'll find this in your booklet, and as we're going through it, you can annotate this diagram. Here we have the afferent arteriole, therefore you should have an arrow pointing towards the glomerulus. On this side, this arteriole is the one that's leading away from it, so it's the efferent arteriole. This knot of blood capillaries, of course, is the glomerulus, and the glomerulus sits inside this C-shaped cup, which is the glomerular capsule. Now, what we have occurring here is glomerular filtration, so the liquid that's caught inside the glomerular capsule is glomerular filtrate. And lastly, as that glomerular filtrate processes through the nephron, it comes to the first part, which is the proximal convoluted tubule. So now we're going to add arrows to our diagram to illustrate what's happening. We can see that blood is under pressure filtration, so blood enters the nephron at the afferent arteriole, and as it's in the knot of blood capillaries at the glomerulus, we undergo that pressure filtration and those small molecules move out, and as that blood flows through, it leaves in the efferent arteriole. So now we've talked about the indiscriminate filtration that's happened, and we now have filtrate in the nephron, which is quite separate from blood. Now, selective reabsorption, as the name suggests, we're choosing what we want to reabsorb, what we want to bring back into the blood from the filtrate. So you'll probably identify the good things, and you've probably realized that glucose and amino acids are the most precious to your body. So thinking about the long process of digestion, we've just made a big effort to digest food and get it into the blood, so we really don't want to lose that. So as soon as we can, we will reabsorb that back into the bloodstream. So the first part of the nephron where we have the opportunity to do that is the proximal convoluted tubule. And these arrows are just signaling that these substances are leaving the nephron and they're going back into the bloodstream. And they are needed because they are essential to functioning of our bodies. They are useful to us. So just to summarize, The proximal convoluted tubule is the location where selective reabsorption happens almost immediately for glucose and amino acids. Most of our mineral salts will also be taken back, and most water. So the mode of transport for these things is by diffusion and active transport. We do not want to excrete these substances, that's undesirable, so we want to take them back in. We can see on this diagram that there are other opportunities to reabsorb those nutrients back into the body. So at the loop of Henle, on the descending limb, going down, some of our water goes back into the blood by osmosis. Now the reason why this happens passively by osmosis is because the water concentration in the nephron is much higher. Now if you think about it, when we underwent filtration at the glomerulus, some of those small molecules that came out was water. So that meant that the blood viscosity was quite high because there wasn't much liquid water there. That is why most of that water goes back into the blood at the proximal convoluted tubule by osmosis, and we get a little bit more happening in the loop of Henle. This is the body again trying to address homeostasis and keep its balance. When we look at the distal convoluted tubule, again we get some selective reabsorption happening here, and this time not most mineral salts but some. So as we're progressing through the nephron structure, the body is starting to address its needs, and at this point in the distal convoluted tubule it knows that it needs more mineral salts such as sodium and chloride ions, and it will actively take these back by active transport. When we get into the collecting duct, this long tube here, again final water is reabsorbed by osmosis back into the bloodstream, and everything that's left over in this nephron is metabolic waste that is going to be excreted from the body. Now, not only do these metabolic wastes get excreted, any excess water and mineral salts that we don't need to maintain our homeostatic mechanisms will be excreted also. Okay, so what we've talked about as a little summary again is we have had glomerular filtration. Blood enters at the afferent arteriole, which has a wide diameter. The blood is filtered under high pressure in the glomerulus, and the blood that leaves goes through the efferent arteriole. After filtration of our small molecules, we will then be into the nephron itself, where our useful substances are reabsorbed. So these peritubular capillaries that wrap around the nephron are in close enough proximity that diffusion, active transport and osmosis can occur, and our useful substances such as mineral salts, water, glucose and amino acids can move back into the bloodstream. And this little diagram here is illustrating what's happening in that process. So here we can see selective reabsorption of sodium, glucose and amino acids. Okay, so here's a summary of selective reabsorption. I'm going to pay attention to the part of the nephron, so this proximal convoluted tubule. This is where most of these good substances are reabsorbed back into the bloodstream. We know that water moves passively by osmosis, so there's no energy involved in that. However, the ions, glucose, amino acids and vitamins, these are all actively taken back into the bloodstream. So this is active transport here, moving against the concentration gradient. Now you'll notice that it's 100% of glucose, amino acids and vitamins that are taken back into the bloodstream, and a significant proportion of ions also. In the loop of Henle, again a significant proportion of water and sodium and chloride ions are taken back, again, water moves by osmosis, which is a passive type of transport here. So in the first half of the nephron, water moves by osmosis, and ions and useful substances move by active transport. As we look at the second half of the nephron, so that's the distal convoluted tubule, you'll notice that the water starts to move actively against the concentration gradient. And this is the first time we've ever talked about water moving actively. So it's moving from a low concentration to a high concentration. And this is the body's way of making sure that your blood has got a good concentration of water inside it. Again, this active transport of water happens in the collecting duct, and this ensures that the body and the urine are at the right concentration of water. Okay, so back to our second diagram, and again we're just going to label each part. Hopefully you should be comfortable labeling these now and you understand what each section is called. The green arrows that I'm drawing here on this diagram are demonstrating that substances are leaving the nephron. So they're leaving the nephron and moving back into the bloodstream, and that of course is what we've discussed: water, glucose, amino acids, sodium and chloride ions. Down in the loop of Henle we've got water and sodium and chloride ions, and at the distal convoluted tubule we have got more water and sodium and chloride ions. And at the collecting duct also. So it would be a good idea at this point to pause the video and just to write down the type of transport that each of these substances undergo to get out of the nephron. Okay, so we've looked at the function of the nephron in its purpose of selective reabsorption, and the reason why it's so good at it is because of these two key features. Because we have these twists and turns, the nephron itself has got quite a long length. So the proximal convoluted tubule, the loop of Henle and the distal convoluted tubule contribute towards that. And we also know that we don't have one large nephron, we've got over 1 million in each kidney. So having lots of them with a large length will enhance the surface area to volume ratio for rapid absorption, reabsorption and filtration. Okay, the next part we're going to be looking at is something called tubular secretion. Now, instead of going from the filtrate into the blood, we're going the other way. Okay, so we are adding things from the blood into the filtrate. So if we look at this cartoon diagram here of the nephron, we are not taking things out of the nephron, we are adding things into the nephron. And the idea is to get rid of what we don't need. So what actually happens here is unwanted substances that are in the bloodstream need to be added to the nephron so that we can get rid of it. And you can see that one of those things are hydrogen ions. You can see that here and up here. Now, we want to get rid of excess hydrogen ions in the bloodstream because we need the pH of the blood to be neutral. By removing hydrogen ions, we're stopping the blood from getting acidic. We also are adding ammonium ions. Okay, so these ammonium ions, that NH4+, that's chemical formula for ammonium, these make the urine slightly alkaline, so hence when we add the hydrogen ions it makes it slightly acidic. So with tubular secretion, we add things to the nephron. So what we add to the nephron at the proximal convoluted tubule are hydrogen ions and ammonium ions. At the distal convoluted tubule, we add urea, a substance called creatine, toxins, any drugs that we've ingested like Panadol and so on, and neurotransmitters that aren't working anymore. Now, all of these just undergo one type of transport, which is active transport. So on our diagram, again in your booklet, we're going to label each part of the nephron. Doing this again is good practice. And this time we are going to add arrows to show what is added to the nephron. So at the proximal convoluted tubule, we have hydrogen and ammonium ions. And what is being added to the nephron in the distal convoluted tubule are urea, drugs and toxins. Okay, so looking at the final urine that's produced, this is the flow from where it starts off in your body to where it ends. So if we look at the urinary tract again, we understand now that the nephrons are located in the kidneys. So they will make their way to the collecting duct. They will then amass in the renal pelvis. They will then flow, that urine will then flow down into the ureters, where it will collect in the bladder before leaving the body via the urethra. Now, the average adult will excrete about 1.5 litres of urine per day, and the concentration of the urine is actually quite low in urea. Most of it is water, and a little bit of it will be ions and other things. Now, it's important to note that in a healthy human, you wouldn't have any glucose or protein in your urine, and if you did, it would be an indicator that your kidneys aren't working properly, or that you may have a disease such as type 1 or type 2 diabetes. So when we look at the filtrate over a 24-hour period, we're going to just look at these numbers for a second. So the amount of filtrate, the volume of filtrate in 1 day is 180 litres of water. Now, that doesn't mean that your body has 180 litres of water. What it's showing you is that this blood is constantly being cycled and filtered to the point where you end up with a volume of about 180 litres of blood going through those kidneys each day. Most of that is reabsorbed back into the bloodstream, and about 1 to 2 litres is excreted as urine depending on your fluid intake. When you look at these data tables, it's important to note that nearly all of the ions are reabsorbed back into the blood. There's hardly any protein at all in the urine, and there's certainly no glucose. Okay, so if these numbers were different, that would indicate to you that there's a problem and this person could have diseased kidneys. Now you'll note here that the final concentration of urea in urine is about 25 grams, and because we started with 53, you may be wondering why 28 grams is reabsorbed. It's not because the blood wants to keep it there, it's because filtration is a dynamic process and blood is constantly flowing by, which is why we need to have continual filtration and continual blood being pumped around the body, so that if blood isn't filtered on the first go around, it has many other opportunities to do so. Now this table is a summary to show you the relationship between the structure and the function of the kidney nephron. I'm presenting this information as a table to you because it's a really good summary that you can use in an extended response question. So please take the time to pause the video and read through each of these points carefully and make sure that you understand how they work. Please note as well the use of key language. So I'm talking about the efferent and afferent arterioles. I've used the word convolutions, and I've talked about reduction in diffusion distances, and I've mentioned things like concentration gradients. Okay, the last thing I want to show you is a summary of the nephron. So from the picture that you drew in the last lesson, it would look something like this. You can either add to it if the piece of paper is big enough, or you might want to stop and redraw it to summarize what's going on here. I would highly recommend that you do so, and you don't have to use these colours. You can select your own colours to make a key, but if you can actually draw this and summarise what's going on without the aid of your notes, that's a strong indication that you have really understood this lesson. If that takes you a few times, that's not a problem. Active engagement with writing your notes is going to be the best way of you remembering this process. Okay, so this is the end of the lesson. Please make sure that you do complete your notes in your booklet and you do read those pages. Now, the next couple of pages, on pages 180 and 181 of your textbook, refer to dysfunctions of the renal system. I need you to go and read that, please. It's an important part of your course, but it hasn't been taught here. You need to be able to read that in your textbook and answer the questions that have been provided to you in your workbook.